Morning. My name is Glenn Browning, and just a few items of housekeeping. Today's session will be recorded and made available on the NCAS website after the session. And if you have any questions, can you put them in the Q&A section at the bottom of your screen? There'll be time for questions at the end of the two presentations. So we'll run from one presentation to the other and then have questions at the end. So today we have an environment theme, and the first speaker is Erica Donner. Erica is the director of the Cooperative Research Centre for Solving Antimicrobial Resistance in Agribusiness, Food and Environments, or the SAAFE-CRC. And she's also a research professor at the Future Industries Institute at the University of South Australia. Has that changed? Is it now the University
Almost.
Yeah. On the verge of changing that title.
Halfway through.
And she's an interdisciplinary environmental scientist specialising in systems-based contaminants analysis, risk assessment and management. A member of the ASTAG group on antimicrobial resistance and co-leads the water, soil and food security theme for the NHMRC Healthy Environments and Lives National Research Network, and is a One Health advisor to the Australian Pathogen Genomics Program, all things that some of you will be familiar with, I'm sure. So thanks, Erica. I'll let you introduce your talk and take us away.
Great. Thanks, Glenn. All right, I'll share my presentation. Okay, is that good? Yep.
That's good.
Okay, looks great. All right. Well, yeah, thanks, Glenn, and thanks very much for the invitation from ANCAS to speak this morning. I'm very happy to have the opportunity to join the webinar today and to share this presentation with you about the environmental dimensions of AMR. I'd like to start by acknowledging that I live and work on Peramunk and Kaurna country in South Australia. And get my slides moving. Not working. Sorry. There we go. Okay, so I'll start by placing this presentation in the context of my day-to-day job, as I know that the environmental dimensions of AMR are not necessarily front and central to many of the people who'll be joining today. So as Glenn said, I work for CRC SAFE, which is a 10-year Commonwealth-funded program that's assisting industries, government and researchers to monitor, assess and mitigate AMR. SAFE's a partner-based organisation, so it really brings together different types of stakeholders and across a range of sectors to help them identify and work and solve their various AMR challenges. So we work across a broad range of sectors, and so this slide shows sort of the breadth of sectors that we work with and relevant sectors to the work that we're doing. And I know a lot of people joining might be from the human health sector, so I guess what I want to do here is just really place it in the context of what the work that we do is relevant to the human health sector and the environments, such as hospital environments and the wastes flowing out of those facilities are relevant to the work that we're doing, but we're largely focused on the food and agribusiness and environmental aspects of AMR. So this is kind of my day-to-day focus and the lens that I'm looking through when I think about One Health AMR day to day. So, and I think it's important to recognise as well that within any one of these sectors, there can be dozens of individual industries and very varied practices and challenges facing them. So yeah, basically the environmental dimensions of AMR though are at the heart of what we do in SAFE. We use a systems-based approaches to address the issues and understand what's going on within any particular production environment or environmental system. So I think a really important thing to recognise is that within all of these different sectors and industries, AMR and disease can look very different. So this is just, a snapshot of what a fungal infection looks like in plants, for example, in this case in wine grapes. And this is the day-to-day reality of the the jobs that people are doing. And so AMS and stewardship looks very different to people in their different contexts across the One Health system. So yeah, really AMR is a multi-sector issue, and so too is antimicrobial stewardship. So we really approach this by recognising that AMR affects lives and livelihoods. It's a multifaceted industry challenge, and it has really broad-reaching health and economic impacts across industries and communities. It's both a food safety and a food security issue. So in addition to being obviously a human health issue and animal health issue, but agricultural productivity is impacted by resistant disease, and that impacts on treatment costs, product losses, and so on. And it's a biosecurity risk, so it can be transferred. I mean, resistant disease and pathogens and the genes and so on can be transferred between different production and food and feed processing systems. And it's actually the environmental dimensions, the waters and things like that, that can play a role in that too. So we do need monitoring and mitigation measures across a range of contexts. And the specific challenges vary by sector, but they include a lack of awareness in many cases and a lack of sector-specific tools and guidelines. So yeah, that's it really, just to introduce that AMS looks and sounds different in different sectors and environments. So why are we really interested in the environmental dimensions of AMR? I think it's becoming increasingly recognised that taking a One Health approach requires us to do just that. So we need to recognise and address the environmental dimensions of AMR if we truly want to take a One Health approach to managing it. And so this has come through over time through the guidance from things like the Global Action Plan released in 2015. It's there in Australia's AMR action plans and strategy. And from the 2015 to our 2020 and beyond strategy, we really moved a long way in having the environment sort of understood and recognised as an important sector that has a role to play in managing and mitigating AMR risks. But Australia's yet to have state action plans that are One Health AMR action plans. Some of them have been in development. But really, being that we're a federated system, we can expect that these state action plans may be needed to achieve significant headway in some areas as well. So as it became recognised that the environmental dimensions of AMR are important, the tripart organisations of the United Nations were joined by the United Nations Environment Programme and became the quadripartite then, with the WHO, the Food and Animal Organization and the World Organization for Animal Health and the United Nations Environment Programme now all joining together to take a coordinated path in addressing and driving forward action to mitigate AMR. So there are many documents that have since come out that help to put this in context. And if you're interested in looking further into any of those, these are some key ones there, including summaries for policymakers and just documents that really spell out all of the ways in which AMR is linked into environmental management and things like water and sanitation. Within our own action plan, you can see that we have here the environmental services sector, as well as agriculture, animal health, food and human health. And they're all interlinked, but we're calling them out saying, you know, each of these they have a role to play. And this is quite advanced in relation to many of the other action plans for AMR across the globe, that we really have this very clear line of environmental action and sector responsibility within our plan. I won't go into the details there, but really we're trying to understand then and bring it in to recognise that the environment plays a role, and how do we integrate this into the system? Well, if you start to unpick it, it looks very complicated. There are many different aspects of environmental management, such as sewage and wastewater treatment that are really important. But really we start to see, once you put the environment in the picture, you start to see that everything's very connected and the things we're doing in one place can be impacting on another. Of course, environmental AMR is also very important because the environment is where a lot of our antibiotics come from in the first place. So antimicrobial resistance itself, of course, is an ancient ecological phenomena. And we know from looking into isolated cave microbiomes and isolated environments that, there is this obviously very long history of having resistance within microbial communities, and it is, of course, their way of dealing with each other. And the vast majority of antibiotics were discovered by screening environmental microbes. So that's an important thing to keep in mind, because when we work in the environmental context, we have this vast natural environmental AMR background that is effectively, well, it's a source of new resistances, but it's also a noise within what we're often most interested in, which is the anthropogenic environmental AMR. And so this is really the changes in microbiomes and resistomes that are occurring because of what we're doing with antimicrobials and other agents that are driving the development of and spread of AMR. And in particular, of course, we're very concerned about multi-drug resistance. And this is just an example of some data we have going down river transects. These are in this case, it's a 132-kilometre river transect from a protected, fairly pristine area of the environment, which is the headwater to the river. And down along that 132-kilometre transect, there are towns, there are farms, there are, different types of wastewater treatment plants, production facilities, and so on, hospitals and the like. And you can see that in the case here, we can use Blatem, which is, a resistance gene, to really show that there are anthropogenic impacts on that river that are being clearly seen in the increase in that particular resistance gene. So this is what we would call anthropogenic environmental AMR. It's not the natural microbiome. We are impacting on what's there. And this is just one gene. Obviously, the whole microbiome is very complex. And so what's going on there, we'll see particular places where there might be downstream of some kind of facility that is leading to an increase in resistance in that area. But this is the kind of ways that we might think about a resistance in the environment. Often we're looking at individual genes and markers and trying to understand what are the sources of environmental pollution, if you like, of resistance in that system. And this is so that we understand where the sources are, so that we might better control and mitigate the releases of resistance into the environment and downstream impacts of that. So it's very important. Obviously, resistance is driven by use and release. In this context, we can see that what we do has an impact on the environment and on the downstream uses of water and so on from that environment. So this is a stewardship issue. When we place all of our sectors within the environmental context, the place and where they're located and how they draw off similar environments and link together becomes very important. So here, just in this graphic, you can see that the different facilities and users of antimicrobials might be quite separate. Some of them will be particularly important, like hospitals and healthcare facilities, for example, the feed mill where antimicrobials might be being added for some uses. And of course, we also use, many fungicides and so on in horticulture and plant production. So we have these different sort of areas that are quite separate within the environment, but here the dots can represent that they're using antimicrobials, but the dots could also be representing just the resistant organisms, pathogens themselves, or the resistance genes and mobile genetic elements, for example, that they're carrying. So the key thing is we are using antimicrobials throughout these systems in many different ways. We have different hotspots. For example, the hospital or somewhere where we know we're really using and relying a lot on antimicrobials. But other things like fungal agent, fungicides, and antifungals might be used more in like plant industries and human health, for example. So the uses are not all obviously similar. They differ, and some things, are not allowed in some contexts, and others might be used really more broadly than we've recognised before. And it's important to start to understand the proximity of these different uses to each other and how they're impacting on the environments around them and on the water and things that they may all be relying on. So this means that environmental AMR becomes important both for the surveillance and for management. So we know from things like the COVID pandemic, you'll have seen how useful it was to monitor wastewater, to understand the emerging risks and new outbreaks and so on, and to understand that within a geographical context. And so it's likely that wastewater surveillance can play a similar role for helping us understand certain things about what's going on within a particular community, be it human or a population of animals, for example. We can use waste as an intelligence source, basically, and we are early in the times of being able to use that very meaningfully for AMR, but certainly we know that we can monitor resistance and various important pathogens and markers in wastewater. So that's one example of the importance of environmental surveillance. But obviously also our wastewater treatment plants act as really critical control points. Australia is in an advantageous position because we have good collection of wastewater and treatment, and in many cases very high levels of treatment. I'm not going to go into that because Claire will speak more around recycled water, for example, which can play an important role in parts of Australia. Many countries are not so lucky, and it's very clear from the evidence around the world that where waste management is poor, be that pharmaceutical waste, coming out of manufacturing facilities for pharmaceuticals, or where the waste collection and wastewater treatment is poor, these are places where we see a high disease burden and really high risks and impacts associated with AMR. So in that context, we can probably do some things slightly differently or better than we are. We're still exploring how that looks in reality, but we're certainly quite well placed compared to many other places in the world. But really, the environmental dimensions, as I've just sort of shown there, they connect sectors and their challenges. So the wastewater is flowing through into treatment plants, back out into different, either in effluents or recycled waters. And the solid waste coming out of those processes is also going back out into our production systems. So understanding what we're putting in is really essential. The better the stewardship upstream, the better the quality of the products and the recycled materials downstream. And it doesn't just disappear. We have to put these things back in somehow into the system. We need water. We can't, it doesn't just flush away to nowhere. It does come back out into the environment. So this is why it's so important to link up our stewardship initiatives and understand things beyond the immediate context of their use. And the reason for that as well is that once we use antimicrobials in a therapeutic context, they don't just all get metabolized or disappear. They do move beyond the point of use in active form. So about 70% on average of the antimicrobials may be up to that might be excreted in unmetabolized and certainly still in an active form. And within the environment, here's an example of tetracycline degradation pathways, for example. There can be really complex degradation pathways as say that compound is out in a water treatment scenario. Things like UV light impacting on it, or just the degradation processes, or the microorganisms breaking down things themselves. These basically create a really wide range of compounds that are then there in the environment. So assessing the impacts of that and understanding really the whole breadth of the challenge and how to address it and management from an environmental management perspective. So if you're a wastewater treatment plant manager, for example, is very, very complex. And one thing that's really interesting and important is that often we know that some of the degradation products of some of these compounds can be more bioactive actually than the parent compound, and they can be toxic and act in various ways on organisms. So this means that in the context of environmental management, we view antimicrobials as contaminants of emerging concern. And we also consider the resistance genes, the mobile genetic elements, and the anthropogenic resistant microorganisms and multi-drug resistance and so on as forms of environmental pollutants. And at the moment, we don't manage them actively. So globally, people are really grappling with how do we actually do that? We know that we need to do the best source control that we can. So that means make sure that everyone using antimicrobials across the whole One Health spectrum are using them judiciously and in the best manner and not having sort of unforeseen consequences of overuse and misuse. And that's why stewardship is so important in all of its contexts. And so we certainly do need more research to develop evidence-based guidelines for things like water quality management and best management practices for different industries to manage on farm and so on as well. So an important thing to note is that presence doesn't equal risk. If we go into any environmental sample, we will find a myriad of resistance genes and organisms. They are not all of equal concern, obviously. There are things that are emerging as, sort of pathogenic, real issues of concern and global health threats that we're very concerned about if we find them in an environment. But even having said that, we really need to do a proper risk assessment and quantitative microbial risk assessments and things like that to actually understand how best to apply our resources and where to intervene. And Claire's going to speak a lot more about that in the next presentation. But it's just really important to recognize that it's very easy to show that there's a lot of resistance, but we really need to obviously delve deeper to understand, is it multi-drug resistance? Is it in a pathogen that we're concerned about? Really, what is the level of risk? And we'll get more into that. So I think this just demonstrates that we've got this really complex microbiome out there. Not some of it is sort of not really coming into contact with humans. It's getting removed in a treatment plant or so on. This is an example to show you that here, for example, these are clinically relevant resistances that these genes are present in the wastewater coming into these 3 treatment plants. You can see this is the water stream here of 3 treatment plants down the columns. And here is in the effluent at the end of the treatment plant. So even without actively setting out to manage antimicrobials and resistance, our treatment plants were already, and this is quite old data now, probably about 8 years old, they are decreasing and removing these things just because of the nature of the treatment processes. But where is it going? A lot of it's ending up in the sludges. This is the solid waste streams coming out of there. So that's important because those things are also getting recycled. And so we just really need to step through and understand once something's been used and has, left a person in feces or whatever and goes into a waste stream, where does that really go? Because if a person's sick, their waste is going somewhere, and our treatment plants and environmental control points are going to be really essential to protect the health of all the other people and animals living in those systems and drawing off water supply and so on. And again, just to state that the natural dynamics of these systems are quite difficult to to get through to understand exactly what's going on when something that we're most interested in might be a pandemic clone or something that of a pathogen that's actually very rare in a system, but we're trying to understand, is that really a risk here when we've got this whole heap of other resistance and things present that we're trying to see the needle in the haystack, and that within an environmental system, there's a lot of temporal dynamics as well. So seasonal, water flows are changing. This is really just to show you that in a freshwater river system across 4 seasons, these heat maps show that there's a real changeover of the dominance of particular genes and markers across those seasons. And these are like particular resistance genes. These are mobile genetic elements carrying resistance genes together, and these are biocide resistance as well. And I'm almost wrapping up, but I just wanted to point out that it is important to recognise as well that we live in a changing environment. The climate crisis is really upon us, and we're seeing it in things like very rapid changes in surface water temperatures, the oceans, and so on. And it's recognised that this poses a serious health threat. Things that climate change impacts from a health perspective include infectious disease. We see, with things like the melting of the permafrost, for example, releasing pathogens that weren't in circulation. We see that things like floods increase certain types of disease outbreaks. It impacts on the biology of disease vectors. We just really have increasing extreme weather events and the things that go with them, floods, droughts, and so on. And all of those situations tend to lead to different and complex types of disease and challenges, which impact on the use of antimicrobials as well as on their efficacy. So we know that landscapes and farming practices are already changing in response to climate change as well. In many cases, people might be needing to grow particular drought-tolerant varieties or something of crops. It can be impacting on their use of fungicides in those systems and so on as well. It's very, very complex, but it's important to recognise that this is sort of another aspect to the environmental dimensions of AMR. Changes in wildlife distributions, migration, and so on will impact on the spread of different pathogens and use of and AMR as well. As we see, there's very clear links, obviously, between birds carrying disease along flight lines along migration routes and things like that. So overall, it's very clear that implementing One Health policy is highly challenging. It's complicated in the environmental context, definitely by pervasive uncertainties and by the multi-sectoral nature of the problem. There are complex interrelatedness of AMR risks and drivers, and there's analytical and governance complexity. Many industry stakeholders have low AMR awareness and understanding. So within the environmental context, we're working nationally with the Australian water sector to take a coordinated approach to understanding and filling knowledge gaps and starting to work towards having guidance and best management practices to manage AMR. But there's certainly a long way to go in bringing this as an issue that's sort of commonly understood. It's still very new to many people in the environmental services and management sector, including within the regulators. And it's really important that we sort of provide the support and step through this to understand what are the environmental drivers and challenges, but also how they're relevant to other sectors, and how important it is to address this effectively in order to support and protect the other sectors that are operating and within environments and relying on clean environments, good water, good waste management, and so on as well. So yeah, I think that's probably the main thing I want to say about that. It is very important to recognise that while AMR and AMS look different in different contexts, the industry appropriate communication is also really needed in all of the different contexts. I meant to say before when I was showing the wine grapes actually that just the words antimicrobial and stewardship, in fact, they apply in all these different contexts. And when we look into it, we see that, well, these industries are doing antimicrobial stewardship as well, but they're never using those words. They don't recognize, fungicides as they don't use the word antifungal or antimicrobial. They might be calling it a crop protection product or something like that, and their version of stewardship might be called integrated pest management or something entirely different. So it's just really we're at a point in our implementation of our One Health policy and action plan in Australia where we still have the different industries learning to speak the same languages or learning that they're actually doing something called antimicrobial stewardship, and starting to engage fully with the conversation. And so, yeah, it's quite an exciting time to bring all of these conversations together and really have a common sort of understanding, use common tools, and be able to have impact across the system. And there's a lot that can come from the human and animal health sectors because they have been working in this space for longer. So, but it also comes from the other angle. So there are good opportunities. So by attending to the environmental dimensions of AMR, of course, there are potentially new therapeutic options hiding there in our environmental samples and systems. Just as we've been getting antibiotics out of soils for a long time, there'll be more to come. It is important to help secure the longer term efficacy of antimicrobials that we have appropriate environmental management in place. This includes within hospital environments, so having the right tools to be able to detect if there's a reservoir of a resistant pathogen or within an environment, be it in a sink or in splashback from a contaminated toilet, wherever it is. There are lots of different technologies and monitoring tools and things that could be coming out of the environmental sector that may be applied in those other environments and vice versa. And environmental AMR is really important that we manage it to decrease the burden of disease across the system. And that's really the best thing we can do to help prevent and control disease outbreaks. And as I said before, water and sanitation is good water and sanitation management is absolutely imperative in that. We can help industries detect and respond to new and re-emerging threats by providing information out of things like waste streams and environmental samples. And we can also use waste and other environmental samples to help provide information on the efficacy of AMS initiatives and interventions. Again, as I was mentioning with the hospital environment, for example, if there's something around decontamination and so on, some of the environmental surveillance technologies may be of benefit there. We can provide new insights and methodologies with such complex microbiomes to work in. A lot of the early leading metagenomics work came out of the environmental microbial ecology communities, and there is still a lot of work being done, for example, on things like target capture metagenomics and things like that, where we need to really get past just looking at heaps of genes in isolation and really understand what is their genomic context, how important are they in that setting. And yeah, it's another whole area where if people are developing technologies like genomics and things that are important for AMR, this is another area where those tools can be applied and use cases can be developed. So with that, I'd just like to acknowledge some of my colleagues at UniSA whose work I was showing in the slides with data, and some of our key partners. And thank you again for the opportunity to present today.
Thanks very much, Erica. For those of you who arrived late, we're not going to stop for questions at the moment. We'll have questions after Claire's talk. So it's a pleasure to introduce Claire Hayward. Claire's a postdoctoral research fellow in the SAAFE-CRC, and her work focuses on developing a Bayesian network model to support the management of AMR risks. The model helps identify critical control points across water systems and food production, providing actionable insights for public health decision makers. Her research addresses complex environmental AMR pathways, particularly in high-risk contexts such as agricultural runoff and wastewater reuse. Claire's broader research is grounded in improving public health outcomes through applied environmental microbiology. Her PhD investigated the role of plumbing systems in healthcare-associated infections, including large-scale surveillance of pathogens in drinking water, the impact of plumbing materials on microbial contamination, and bioaerosol generation from hand-washing basins. She's a qualified environmental health officer and is committed to bridging science and practice to inform effective real-world solutions. So take it away, Claire. Thank you.
Amazing. Thank you so much for that introduction, and thank you guys for having me to speak today. As Erica mentioned, my presentation will be going a bit more into the risk assessment aspects of our work in environmental AMR, and I'm particularly looking at water, waste, and food and how those systems connect. So for today's presentation, I'll be recapping very briefly about why environmental AMR matters, some stewardship blind spots in our work, how we're reusing water and biosolids, particularly in agriculture, and what that looks like from a risk assessment perspective, and then introducing some of our systems thinking approaches and models with implications and what some next steps are. So following on from Erica's introduction into environmental AMR, I wanted to bring us a little bit closer into some of the practical exposure pathways that link these environmental systems to some clinical and public health risks. So one of the key concerns for us is how AMR genes and organisms may enter, persist in these water and waste systems. These exposures can often occur well outside clinical settings, but can have significant downstream effects, particularly when we look at reclaimed water or biosolids and how they're used in food production and in surface waters. So in this example on the screen, it starts with human antibiotic use, which is taken and then excreted out through the sewage system. This sewage is then treated in a wastewater treatment plant, and this treated effluent might be discharged into a receiving environment. This receiving water may be then used in agriculture for irrigation purposes, which may result in contaminated produce. If this produce is consumed raw or minimally processed, this may result in a human exposure and then a subsequent illness, thus increasing the antibiotic use and then completing this cycle over again. This environmental interface is where my work is focused, understanding these potential transmission routes, current management approaches, and maybe identifying some critical control points that can support safe reuse and manage this AMR risk. So we often talk about antimicrobial stewardship in terms of what's happening here at the point of prescription and clinical governance. And there's some growing evidence demonstrating environmental contamination in the hospital, which is actually where my PhD work was focused. And both of those aspects are critically important. But when we map those systems across the whole AMR pathway, we can see that much of this environmental system is left out or just perhaps not well understood. So we've mentioned wastewater treatment plants a bunch. These aren't designed necessarily or validated to remove AMR genes or organisms specifically. They're primarily built to meet conventional chemical and nutrient removal targets. However, it's just a bit unclear about how these systems perform when we consider biological risk endpoints like AMR. Similarly, there are limited or no discharge limits for AMR or their chemical stressors like antibiotics that may be contributing to this resistance selection in those receiving environments I mentioned earlier. A number of antibiotics have been added to the European Union Water Framework Directive watch list. However, our Australian drinking water guidelines don't list specific antimicrobials, and monitoring in these receiving waters is done sporadically, with no maximum permissible levels of antimicrobials at the moment. So this means with even really great prescribing practices and strong infection control at the clinical point and in hospitals, these organisms can continue to circulate through these systems through water, soil, and food with perhaps minimal oversight or intervention. So one of the environmental contexts in my research focuses on water, how we use it, how we reuse it, and how this pathway connects multiple sectors across the One Health spectrum. As this heat map on the screen shows, Australia is the driest inhabited continent on Earth, and I'm in Adelaide at the moment, so the driest state in the driest continent. However, average annual rainfall is low and very variable, with large areas receiving less than 250 mils per year. Add to that climate change, which Erica touched on, prolonged droughts like the Millennium Drought, and growing pressure from urbanisation and agriculture, it's clear why water security is a critical national issue. And recycled water provides a reliable, climate-resilient supply for agriculture and industry, and even has some potable reuse applications as well. It helps close this loop and reduces the pressure on the rivers and groundwater, and helps support food production year-round, which is really important. As we try to scale up these reuse schemes across the country, it's really important that we ensure that they're not introducing new public health risks, like AMR, for example, that haven't traditionally been part of this conversation. So water reuse supports food security, reduces the reliance on those freshwater sources, and diverts treated wastewater from discharge into a beneficial use, which is fantastic. It also fits within circular economy goals, especially with its nutrient recycling potential. But with these benefits, AMR risks are not well understood. For example, the Australian guidelines for water recycling specify health-based targets for bacteria, protozoa, and viruses in recycled water, particularly for water that's used for irrigation of commercial food crops that are eaten raw. To achieve these targets, we use a multiple barrier approach, meaning the treatment must use a combination of barriers. For example, it's activated sludge, UV, filtration membranes, and chlorination. And this approach ensures that even if one barrier were to fail, there are multiple others as backup to ensure that this recycled water reaches health-based targets, which is fantastic. While recycled water is treated to meet these microbial and nutritional standards, it's not validated for AMR necessarily. AMR genes and organisms may be able to persist, and where that water is used to irrigate raw eaten produce crops like our leafy greens or root veggies, this is how they may enter the food chain. Our work is trying to understand how we can optimize these existing treatment barriers to address this potential risk, which is just one piece of a very complex puzzle. We need to consider the wider system. In agriculture, this soil may run off into waterways. These bacteria might be uptaken into the soil microbiomes, and there's ongoing selection pressure from chemicals that are used throughout the agricultural cycle, which Erica's also touched on. Without AMR-specific guidelines or validation, we risk solving one environmental problem, which is water security, while potentially exacerbating another. Reuse isn't optional in many regions, but understanding and managing this potential AMR risk within it is essential. So when we look at recycled water in Australia, agriculture is by far the largest user, accounting for about 80% of all recycled water use. One of the best known examples here in SA is the Virginia Pipeline Scheme just north of Adelaide, which supplies over 300 growers with recycled water. We also use this in McLaren Vale's vineyards and supporting our key export industry in South Australia. In urban areas, we have dual reticulation systems, sometimes called purple pipe schemes, like in Mawson Lakes, which is where we're working today in Adelaide, and throughout Sydney as well, where tens of thousands of residents use recycled water for non-potable purposes like garden watering and toilet flushing. We've also seen potable reuse in action in Western Australia, where the recycled water is treated and injected back into aquifers to supplement drinking supplies through groundwater replenishment. That's also happening in South Australia as well. And finally, public green spaces like sports fields to city parklands, like those in the Adelaide CBD as well as surrounding parklands, heavily rely on this recycled water for irrigation. So my point here is that reuse throughout Australia is very widespread. is essential and it's already working really well. But as we continue to grow and expand these systems, we need to make sure that we're understanding the potential of AMR risks and manage them as a part of our existing system. Erica also touched on that we use biosolids in agriculture as another byproduct of wastewater treatment. And we also use animal manures as well as a cornerstone of that circular nutrition management as well. So these both manure and biosolids return valuable nitrogen, phosphorus, and organic matter back into the soil. And this is great to reduce the need for synthetic fertilizers. It's also quite a cost-effective solution and can improve soil health. In Australia here, we've got nearly 85% of biosolids may be beneficially reused. And again, 4 to 7 million tonnes of manure is produced annually. So these biosolids and manures may contain residual antimicrobials from use both in the animal sector and in the human clinical sector through the wastewater treatment plant, for example. So that's the chemical stresses, the genes and the bacteria. And once they're applied to the land, these elements may persist over time. And this is something we're not too sure about. And like water reuse, this practice isn't going away, and nor should it. The environmental and economic benefits are really clear, but we do need to ensure that we're managing this organic waste so it isn't overlooked as, again, a potential risk of transmission. What we're highlighting here is that we're not in opposition to it, but we're just trying to think of it from a systems, One Health perspective. We need frameworks that account for these potential AMR risks alongside maintaining those nutrient recovery goals so that we can maximise both the benefits of these practices without introducing potential blind spots. So when we look at applying manures and biosolids, it can provide a range of well-known agronomic benefits. They return those essential nutrients like nitrogen to the soil, but importantly, they feed that soil community. A diverse and really active microbiome in these settings can support plant growth in multiple ways. It increases the nutrient availability, provides a great soil structure, and it can even work to suppress plant pathogens through mechanisms like microbial competition and antibiosis. So these processes are really foundational to making sure that we have a healthy crop system, and are key reasons why these alternatives are used so widely, particularly in low input or circular farming systems. However, when the same fields are repeatedly exposed to these organic amendments containing potential AMR residues, genes, and bacteria, especially in systems where the same crop is grown over and over, season after season, we can begin to see a bit of a shift in that beneficial microbial balance. Instead of a really diverse community that can support the plant health, we may be seeing resistant plant pathogens dominate or beneficial species that are becoming suppressed in that soil environment. These aren't just a theoretical concern. We know that genes and residues have been shown to persist in this soil over time, and in some cases, they may be contributing to the selective pressure that is able to disrupt this microbial ecology that's happening. This could have potential knock-on effects, not just for food safety and human exposure, but also for the plant health itself, potentially reducing the resilience of these cropping systems. So again, it's not just about abandoning reuse in general and going back to those synthetic fertilizers. It's about ensuring that we understand how these products are used and how we can potentially minimize the AMR risks and then boost the agronomic benefits as well. So this involves conducting appropriate cost benefits analysis so we can balance both of these outcomes and optimize the existing thorough engineering systems. Initially, it might be a choice of relevant behavioural changes, like understanding how repeated same crop planting season over season is influencing these, alongside the more physical engineering solutions. Like the multiple barrier approach we had in the water reuse example I mentioned earlier, it's better understanding and stewardship of AMR throughout these reuse chains, both the water and the biosolids, is really essential to understanding and addressing these risks so we can continue these reuse practices that are already so beneficial for us. So when it comes to managing the risks of both water and biosolid reuse, especially in applications like agriculture, we often rely on fit-for-purpose frameworks. These are designed to align with the level of treatment or those health-based targets I mentioned earlier with the intended end use. For example, crops that are eaten raw or reused water that might come into direct human contact might require more robust treatment exposure controls. That might mean a higher level of disinfection, that multiple barrier approach, or site controls like changing practices like drip irrigation to try and reduce that exposure. So that model sounds great in theory, but in practice, effective risk management often depends on the end user consistently applying these best practices. There's only so much we can do from a guideline or regulatory perspective, and that's where it might become more fragile. For AMR, this fragility becomes even more pronounced. So traditional risk assessments weren't designed to account for things like gene transfer, co-AMR selection by other chemicals, not just the antibiotic drugs themselves, and post-treatment regrowth of these resistant organisms. So these complexities don't always fit into the current validation frameworks, but we might be able to adjust and amend them accordingly. So while we have the right aspirations on paper, managing this risk in real world reuse scenarios requires us to update these tools. We need better guidance and stronger support to make sure that on-ground implementation is done so effectively. So these risks don't just stop at the field. Again, we've emphasised this loop over and over. When it rains, the runoff from the land may be integrated back into the receiving environment, where this water is then used for crop irrigation, and that cycle continues over and over. So this circularity reinforces why we need to think beyond the single point of stewardship and consider the full environmental system when we're tackling AMR. So similarly, we've been looking at AMR through a clinical lens traditionally, considering how the recycled water and biosolids might introduce resistant organisms into the environment with downstream human health effects. But when we zoom out, we can see that the system is far more complex, and Erica had a few similar diagrams in her presentation. This diagram is a causal loop model showing how AMR can spread and evolve across multiple interconnected environments. So that included the agricultural land and wastewater systems, in animal production, surface water and composting sites, and a few of these loops are included here. So these environments are often viewed and managed in isolation. But in reality, as we can see here, that they're deeply connected. Each node, or blue label here, can act as both a reservoir and a potential transmission route. For example, the resistant bacteria might develop in livestock or in soil and spread via manure to crops or enter surface water during those rain events, and eventually cycle back to humans through food, water, or direct environmental contact. Importantly, many of these pathways are also direct routes of human exposure. So for example, people that are working in these settings might have a more direct exposure route. So when we look at this system, it's really clear to see that we're not dealing with one linear exposure pathway. We're dealing with complex feedback loops, multiple sectors, and really siloed data sets. Each piece of this puzzle has its own assumptions, its own monitoring processes, and its own risk frameworks. So this is where this type of network modelling becomes a really valuable tool. It allows us to combine these fragmented data sets that sit within each of these sectors, water quality and clinical surveillance, and combine that with our expert knowledge in environmental AMR and uncertainty to map out these critical exposure pathways and identify some potential intervention points. Rather than simplifying the system too far, these models help us really visualize and quantify that complexity in a way that supports that cross-sector risk communication, again, that Erica touched on, and making sure we have evidence-based decision-making before we introduce a new guideline or regulation. So in short, what these systems and models do is help us ask, where can we act? Where does that acting matter most? And how can we make smarter choices without oversimplifying the system we're trying to manage? So when we translate environmental AMR into something that's clinically relevant again, what we've learned is that water reuse and manure applications and environmental microbiology is where AMR genes and resistant organisms may persist, and they may be doing so in systems that we're not monitoring closely at the moment, like in these soils, wastewater, and recycled irrigation water. But these aren't just risks to the environmental setting. as we've mentioned time and time again, they become clinical risks when these resistant organisms make their way into our food and water or into our air, particularly concerning in settings where we have immunocompromised or exposed occupations. For example, when the leafy greens are irrigated with that treated water and are consumed raw, or when the runoff from biosolid treated fields enter our drinking water sources like fresh water. These potential exposure pathways are what are typically unmeasured when we look at solely a hospital or clinical AMR surveillance perspective. We also need to know that resistance genes can persist long after the antimicrobial pressure is gone, particularly when we consider receiving environments that have soils and sediments that can accumulate this over time. And these long-term risks are currently undermined or not addressed in our stewardship goals. So by building stronger connections between the environmental data and clinical endpoints, like infection rates and resistant patterns, we can design a more proactive and again, system-based intervention guideline. So the goal of all of this is not to alarm, but to better integrate environmental evidence into how we understand, monitor, and manage these AMR risks around the whole One Health spectrum, because they're all connected. So to wrap up, I just want to touch on where we go from here. So we know that environmental AMR risks exist across those interconnected systems through wastewater and agriculture to food and our water. It's also important to stress that these systems are not unmanaged. They already exist within well-established risk frameworks in place to manage these microbiological risks, particularly in water reuse and biosolids, and a huge amount of effort has gone into making sure that those systems are safe. That said, AMR introduces a number of new dimensions, again, things like gene transfer, co-selection, and regrowth that aren't always captured in our current validation processes. So that doesn't mean that system is unsafe, but it means that we need to keep evolving our risk assessments and tools over time, and doing so with an evidence base to understand where additional safeguards are needed and which systems are already doing a great job. So now that we know we need a more integrated view of risks, one that includes AMR fit for purpose frameworks and links data sets with clinical relevance and supports decision making on in real world exposure. And so that's what's already underway through SAAFE-CRC. We're taking a national coordinated approach, bringing together the researchers, utilities, regulators, and end users to understand where AMR fits within their systems and how we can best manage it. So finally, I just want to acknowledge the research team that's working with me on this risk assessment project and some of our research partners undertaking this project as well. Thank you.
Thanks very much, Claire. We have a couple of questions on the Q&A. The first from Nadine. What do you think are currently the biggest gaps in managing the environmental risks due to human healthcare? I think it was directed at Erica, but I think we could have either of you talk to that.
I'm happy for Erica to take that one if she's still around. Oh, there she is.
Here. Sorry, so biggest risks in the human health?
The biggest gaps in managing environmental risks due to human healthcare.
Okay, good question. So probably, did you say Nadine? She's probably the perfect person to answer that. I should ask her. If it's the Nadine I'm thinking of. So I'm aware that maybe there are things around waste disposal of unused antibiotics and things like that that are perhaps things maybe still being put down into the wastewater system that perhaps could be diverted or otherwise managed. And yeah, I guess this could differ in different facilities and settings. So I can't really comment on that, but I'm pretty sure that there will be, if we really look through the uses and management and pathways back to the environment, that there are probably some things to address there, and that's one of them that comes to mind. But I probably am not really across all of it in detail enough to pick others from within a hospital. setting. But I think that just in terms of understanding like how much reservoir there can be around just around things like the different washing facilities, basins, and so on. Just how much things can hang around in like drains and so on. Claire's research obviously on is there on that, but things like premise plumbing and how they're managed in hospitals and other healthcare settings, particularly older or extended facilities that have got new wings and things, their water quality can vary very vastly, and you never quite know unless you're looking, and it takes time and money to look. So often we don't know. But that's not that's not really a stewardship, that's an environmental management question. Claire, anything to add?
I was going to say the first thing that came to my mind is my PhD work looked at the hospital environment, and there's quite a lot of information out there about prescribing practices when we look at antibiotics, particularly about dosage and that kind of thing, which is fantastic. And what I saw during the COVID pandemic is people were using a lot of sanitizers and other alternative antimicrobials, which is great, but there was a lot less understanding or information about how those antibiotic alternatives may also co-select for antibiotic resistance. So, and there was also a lot less, the guidelines weren't as strict about how we're using those products. So a lot of times people, particularly in healthcare settings like aged care or healthcare in the home, people were using them at dilutions that aren't appropriate, and that sub-inhibitory concentration might be adding an additional pressure that's selecting for AMR. So I guess it's learning from the guidelines we've put out there for antibiotic use and then applying those learnings to the AMR antimicrobial alternatives that we've started using over the last few years.
Thank you. Kaz, you've got your hand up. Are you going to continue that theme?
No, I want to talk about something else which I really picked up on you with Erica when you talked about the fact that I remember when I went over in Adelaide and we had that great One Health meeting, and I was just blown away by what the wine growers are doing, which is effectively incredible antifungal stewardship programs. And I was so excited. I really want that guy to come and talk to us because it was amazing. And we actually know from social science that the word AMR should not be used. Antimicrobial resistance AMR shouldn't actually be used in any policy because people, as you say, don't understand it. So I think the policy, and we've also done some work with the Mark Davies, where we did also a social science with diving deep into understanding whether One Health is really very effectively addressed in our policy documents, and it actually isn't. It's sort of a bit sort of virtual or sort of vaporware, I should say. So I actually do think we have a real issue with our wording, our how we're trying to get it across, and hanging out on AMR as a thing. But actually, I think it's actually about environmental safety, animal safety, and human safety. And I just think that we need to work together to work out how we communicate this problem, because it hasn't got any better. And I've been in this space for like 25 years in terms of understanding for non-clinical people, or, I just, you're around it all the time. So I'd be really interested in your thoughts here.
No, I mean, I totally agree. Like, it is a big, there's a gaping sort of, chasm between in the language and understanding what AMR is and means. So I think, though, we just have to meet people where they're at. And so it really is like, I mean, we don't necessarily, even with our work, sometimes, there'll be people who are working directly with farmers who are like the consultants supporting them, or the extension officers from the, peak bodies and things like that. So who they'll be doing their education at grower days or something on farm, and they're just using the appropriate language, and I think it's just putting the stewardship into the actual day-to-day context that counts for those people, and not trying to introduce necessarily new terms and language, but really just work with the the terms that work there, and but but understand it and build out the system so that from a policy and academic perspective, we're starting to connect things up. But then, yeah, just not expecting that everyone meets us at our terminology, but really that we take it out to them in the language that counts. I think that's important. I don't know if that really answers, but yeah.
Carolyn, have we got a bit more time? We've got a few questions in the Q&A.
Maybe one more question, and then we might need to wrap up after that.
There's a couple of locals, so I'll jump to the international question from John Prescott from Canada. What do each of you think is the highest current environmental risk for human health that needs to be addressed? Hospital sewage waste, municipal waste, recycled water, farm animal waste. Which do you think is the biggest risk?
I think it's always going to depend on the local context. So, yeah, I mean, I think in countries where the wastewater collection and management is poor, that's going to be absolutely critical. And so, like, breaking those transmission paths, and it can be very difficult, obviously, in places where you've got sort of informal settlements or not good sewage collection and things and treatment. So there, I think that's absolutely fundamental because the pathways can be close back to people, but that obviously differs depending on the country status and so on. I think they're all important, but the human wastewater and the links back to where we're using critical antibiotics and things is probably particularly important, I guess. Yeah, I'm going to go with that.
And Claire, do you have a critical control?
Yeah, in terms of the wastewater, what we've seen is it's really dependent on where the system is placed. Not all wastewater treatment plants have a hospital or an abattoir, which we might consider being a higher risk discharger. So that is a concern in those specific contexts where we do have a large amount of hospital wastewater that would have a higher amount of those chemical stresses and potentially resistant organisms. One potentially more concerning point from my point of view is the farm animal waste, because we don't really have as much information about what's happening in those practices, nor is there currently as robust treatment or intervention guidelines. In the wastewater perspective, it's really well managed, and it's really kind of easier to access that data or understand how that waste is moving through a wastewater treatment plant. But understanding what's happening in practice when we're using the farm manure as a potential source of fertilizer is really important. That's something that we'll be looking into to understand if it's poultry, pig, or cow manure, for example. What are those practices looking like, and what are people doing on the ground to kind of either manage this risk, or how are they using those sources? So I hope that answers, or kind of tries to answer that question.
I guess the critical thing with agriculture is that we don't tend to use the high importance antimicrobials in agriculture. So some of the risks are going to be different in terms of which drug resistance is being selected for. There are a few questions in the Q&A. Carolyn, can we get those to Erica and Claire to respond?
Yes, sure. And I suppose
from
also depending on if Erica and Claire are happy, if those people possibly might want to contact them directly if they want to speak in more detail about those. But yeah, certainly I can get those questions to you.
Okay, actually there's 3 there, but you've captured them, have you, Carolyn?
Yeah, I've got them.
Okay, so we're starting to lose a bit of the audience anyway because we've run over time. So I'd just like to thank Erica and Claire for very interesting presentations this morning. Thank you. As you can gather from the interest, there's a lot of people interested in what you've been saying.
Great.
Thank you very much.
Thanks again.
Thanks,
and just a reminder, the recording will be available after the session, and the next NCAS seminar will be on Wednesday, the 20th of August, with a theme on antimicrobial stewardship in animal health. Thank you.
Thank you. Thanks, everyone. Thanks so much for a great session.
Thank you. Bye. Okay.